Welcome back. We're now going to learn about the abdominal accessory organs. Specifically, we're going to learn about the liver, and tucked underneath the right side of the liver, we have the gallbladder. It's attached to the liver through some bile ducts that drain from the liver. And then there is a common bile duct that will then go through the head of the pancreas. So the pancreas is actually an organ that is retroperitoneal. It sits behind the stomach. But notice that the pancreas is kind of tucked into the C-shaped portion of the first part of the small intestine, the duodenum. So that after the pyloric sphincter, the small intestine has to dive retroperitoneal so it can get, excuse me, secretions from the pancreas as well as from the bile duct itself. So let's get started with the liver. The liver is the largest organ inside your body. Most of it is tucked up under your rib cage on the right side of your chest, but as you can see, it does cross over the midline to the right side as well. When we look at what's going in and out of the liver, from the aorta, we have a hepatic artery which contains oxygen-rich blood, but this is only 20% of the blood supply going into the liver. 80% of the blood supply comes from the hepatic portal vein. Yes, a vein brings 80% of the blood supply to the liver. This is very nutrient-rich blood, but oxygen-poor. I've talked about this before, but I'll mention it again on the next slide if you haven't heard about this already. As the liver makes bile, that is going to leave in bile ducts, which will then take that to the gastrointestinal tract. Specifically, it'll be draining into the first part of the small intestine, the duodenum. And then blood will exit the liver through a hepatic vein, which will then drain into the inferior vena cava. And so this is the last vein below the level of the diaphragm, and then the inferior vena cava punctures the diaphragm and then dumps directly into the right atrium. No other veins will drain into the inferior vena cava after the hepatic vein drains. So to review the hepatic portal system, we have arterial blood that's entering into our stomach and intestines. And from inside the lumen of the stomach and intestine, we have a capillary bed that is absorbing nutrients as well as possible toxins and drugs that may have been consumed. And so that's in the capillary bed and is drained into the venous system. Now, we don't want that venous blood to go directly to the heart where it could cause you to have your heart stop, or for it then to go throughout your entire body from your heart where it could kill you. So we have a portal system. Remember, a portal system is where venous blood goes from one organ to another organ before it goes to the heart. And in this case, it's a hepatic portal system, meaning this vein, the veins that are draining the stomach, the small intestine, and the large intestine are all going to travel to the liver through the hepatic portal vein. And so this is where 80% of the blood entering the liver is coming from. This is nutrient-rich blood, oxygen-poor blood, but possibly containing toxins. And so this blood enters the liver where it has in the liver cells a second capillary bed between the liver cells, and the liver cells are going to be detoxifying this blood as well as absorbing nutrients and doing other things. And then this clean blood will exit the liver through hepatic veins right here into the inferior vena cava. All right. So when we look at the microanatomy of the liver, it is composed of what are called liver lobules. And so here's a photomicrograph of actual liver. And you can see these liver lobules in the center of which is a little venous thing of each of them. And they're all kind of shaped like stop signs, okay? And when you look at it, every corner of the stop sign has the same three structures, okay? We have a branch of the hepatic artery bringing blood in. We have a branch of the hepatic portal vein bringing blood in. And what is going to happen is that blood is going to co-mingle and travel towards the center of the liver lobule. And notice when it travels there, the hepatocytes are on both sides of this capillary bed. And so hepatocytes are only one or two cells on each side of it. And then blood travels towards the center of this liver lobule, and then eventually these will coalesce to the hepatic vein going out. Now, additionally, these hepatocytes are going to be making bile, and the bile ducts are going to be carrying bile out of the liver lobule. So it's the only thing that's going the opposite direction. So here is an electron micrograph of a liver cells, in this case of a liver. Here's that central vein area, and here the liver cells are green. I don't know why they made them green, but you can see all the little capillaries between the liver cells where the work would be being done for detoxification. So the functions of all these liver cells: detoxify blood, number one, most important thing you would think. In reality, not the most important thing, because most of us don't go around eating poisons all the time, but it's always there if you need it. So yes, it's going to have the necessary enzymes to detoxify your blood. But the liver does a whole slew of important things. Temporary storage of iron. Okay, so when you are breaking down your red blood cells after 120 days, if you have enough iron, you can store extra iron here. If, on the other hand, if you're a woman in your childbearing years, you probably don't have extra iron, and so that iron gets reused right away in your red bone marrow for making new erythrocytes. We try to have a 48-hour storage of glucose, the storage form of glycogen. Remember, glycogen is just a long chain of glucose molecule. Our fat-soluble vitamins are stored in our liver, so vitamins A, D, E, and K. Additionally, vitamin B12 is stored in our liver. If we are having a low blood sugar, our liver can release glucose if it needs. It can take those glycogen stores and break up the glycogen and release some glucose off of it. It can also make new glucose molecules if needed. Remember how we said most of the proteins in your body are made in the liver? Well, got work to do that. And so albumin, more than 50% of your plasma proteins were albumin. We talked about immune globulins, so all those antibodies. Remember when we talked about your blood clotting and with platelets, all those clotting factors in that coagulation cascade, all of those that are floating around in your blood in inactive forms, they are made in your liver, okay? And when we talked about non-specific immunity and we talked about the complement cascade, the end result of which was that membrane attack complex, well, those components are also made in your liver. So now you see how important your liver really is, now that we've learned about all those other systems in your body. And did you know you don't need to eat any foods that contain cholesterol, because your liver will make all the cholesterol you need. And it also tries to regulate your cholesterol levels by making something called bile salts, which are in the bile that your liver releases. So Your bile also contains a breakdown product of your red blood cells, something called bilirubin. And that combines with bile salts and the cholesterol and the water, and that's what actually makes that green substance that is known as bile. So bile, when it's made by the liver, it's made 24/7, and it will drain directly from the liver down through the bile ducts and go through into the small intestine. However, at night you're not eating, so there is a little sphincter here at the small intestine that will be closed, and so then bile will go into your gallbladder and be stored for later use. So the gallbladder is tucked under the right side of the liver. And so this is why, another reason why you needed to know that first portion of the small intestine, the duodenum, because tucked in there, that is where the bile duct will enter. And notice the bile duct enters along with the duct containing secretions from the pancreas. They join at the same time, and that's crucial. And that is because bile emulsifies fat. And you're going, well, that's great, but I don't know what that means. What that means is if you have a big fat globule and you put bile salts with it, that big fat globule is made into smaller things. It's kind of the same thing that happens when you take a bottle of salad dressing and you shake it up and you see all the smaller little globules of fat mixed up among the vinegar, because the vinegar is water-based. Okay, that is a mechanical digestion of the fat blob. That's what bile is doing. It's mechanically digesting it. So now, instead of having the surface area of one big fat globule, you can see all the green surface area of all these smaller fat globules where the bile salts are. And so now if we ever get an enzyme that will act on lipids, we have a much bigger surface area. Now, if the bile gets super saturated, especially with cholesterol, we can get gallstones, and it's very common. And you can have a plethora of small gallstones, or you can get really huge gallstones. Doesn't make a difference. The small gallstones, unfortunately, can get into the little ducts and completely block them. Okay, so it does make a difference because the small ones can make you really, really sick by blocking the bile ducts, whereas the big ones can just give you a gallbladder attack by blocking the gallbladder. But they are too big to get into the bile ducts themselves. So moving on to the third organ. So the third organ, the pancreas, actually sits behind the stomach. So the stomach has been pulled up in this case so you can actually see the pancreas. And the pancreas has two functions. We've already talked about the endocrine function of the pancreas, and that was with these islets of Langerhans, where we had different cell types, and we talked about the alpha glucagon and the beta cell secreting insulin into the bloodstream. So in addition to that, we have an where we actually have cells here, which are going to be secreting digestive enzymes into ducts. And then the cells that line the ducts themselves also secrete substances. And all of these secretions will travel down these ducts and join up with the bile from the common bile duct, which is what this green thing is. Together at the same time when this sphincter is open into the duodenum. Okay. And what we call this mixture of substances coming from the pancreas, that is called pancreatic juice. Okay, so the pancreatic duct joins with the bile duct inside the bile, and the pancreatic juice are released at the same time in the same location into the duodenum of the small intestine. So here's what's in pancreatic over here on the left side. Bicarbonate ions and water. So bicarbonate, the whole point of that is bicarbonate has a has a negative charge. So it has a very high pH. And the point of that is to neutralize the very acidic from the stomach. So we won't have an acidic chyme eating up the small intestine. Because notice, I've never said anything about small intestine cells secreting a mucus layer to protect the mucosa. So we have sodium bicarb coming from the pancreas to protect it. Now, on the digestive enzyme side, we have stuff for chemical digestive. We have a whole bunch of inactives that are proteases. So remember, they're going to be digesting proteins to give us amino acids. So for this course, you don't need to know trypsinogen becomes trypsin, chymotrypsinogen becomes chymotrypsin, etc. Just call them inactive proteases. So when they're active, they're proteases, going to be digesting proteins. I'll be showing you in the next video. In addition, we have some active enzymes. Why? Because what they're active against is not present in the pancreas normally. We have amylase, and we've already learned from our salivary glands that amylase digests carbohydrates. And so now carbohydrates eventually are going to be broken down and give us those monomers, specifically glucose, which can be absorbed. Can also give us fructose and galactose. And then the other one that we haven't talked about before is lipase. Look at the word. Can you see the word lipid in lipase? So lipase is going to be digesting lipids into glycerol and fatty acids so we can absorb the fatty acids. Now that's going to be a little bit complicated, so pay attention when we get there. So here is the summary for the accessory organs. So bile coming from the liver can be stored in the gallbladder or can join up with pancreatic juices to try to neutralize the acid chyme, and then we'll be able to digest proteins, carbohydrates, lipids, and I haven't talked about it, but we're also going to be able to be digesting nucleic acids. So how do we regulate these digestive secretions? Well, this is actually fascinating. It's controlled by parasympathetic nervous system as well as digestive hormones. So we've been talking about these things as enzymes, but anytime an enzyme goes into your bloodstream and has to travel to a different organ, it's a hormone by definition. Okay. So as you know, especially if you've ever heard of the Pavlov dog experiments, anytime you look, smell, and think of food, we get gastric secretions. And here's the thing, when we eat protein, our stomach produces something called gastrin. So I know I haven't talked about gastrin before, but when we eat protein specifically, we're going to make gastrin. And what gastrin does is stimulates our gastric glands to make pepsin. Well, it stimulates the gastric glands, which make hydrochloric acid and pepsinogen, and then the hydrochloric acid converts to pepsinogen, and we get pepsin. And as you recall, the pepsin breaks down the proteins that gave us the gastrin, that gave us the pepsin. So we've done a lovely little circle, haven't we? What started the whole process broke down what started it. A little auto-regulation going on. Here's another one, and here is the picture, and I've colorized it. So the reason why I put pepsin up here in blue is because that's what it's showing it's here. And so, yes, the pepsin's going in your bloodstream, and yes, the pepsin's coming back. So yeah, pepsin acts as a hormone, as well as it's got paracrine function, as well as endocrine function. All right. Now, when dietary protein and fat enter your small intestine, those small intestine cells produce something called cholecystokinin, or in this case, just abbreviated as CCK. And CCK is what is being shown by the purple arrows here. So we have CCK leaving the small intestine and going in the bloodstream. And now what CCK is going to do is CCK is going to go back up to the liver and the gallbladder and make the liver produce bile. And remember, bile can come back in the small intestine, and it also makes the gallbladder contract. So whatever bile was in the gallbladder is going to contract. And so now we are going to have bile going into the small intestine as a result of fat being in the small intestine. Bile will enter the small intestine. And so dietary fat will be emulsified. So do you see, fat in the small intestine gives us CCK, which makes bile go in the small intestine, which will then mechanically digest that fat. Oh, but there's more. Because in addition to this fat giving us CCK, hydrochloric acid in the small intestine gives us this green substance called secretin, which goes in the bloodstream, and secretin and CCK together are going to act on the pancreas to give us pancreatic juices. And as you recall from pancreatic juices, one of the things in pancreatic juice was the bicarbonate ions, yes, which can buffer it. But in this case, because I was talking about CCK, the one thing I am interested in was the lipase that was going to break down the lipids. So not only have we mechanically digest the lipids, but now we are chemically digesting the lipids. So what a tangle weave we have web. Oh, and let me give you a piece of trivia about secretin. Secretin in 1902 was the very first hormone that was ever discovered. Yes, by the same lab at McGill University that had to what Pavlov moved to with his dogs. So fascinating stuff there. So thank you so much for your hard work. I know this was a little confusing. So just kind of follow the hormones and it kind of all makes sense. I'll see you soon as we finish out. I'm going to review the digestion and absorption first, and then I will, so we got 2 more videos, and then I'll finish the large intestine.